I've been asked to introduce this panel today about rare breakthroughs in research and care. And we'll say we have a distinguished panel here and really want to get to the meat of the program. It's going to be moderated by Dr. Shuster, who happens to be NORD's vice chairman of the board, and also the president of Keck Graduate Institute. And we are fortunate to have Dr. Donald Kohn, who is professor of the Human Genome Medicine Project at UCLA, and Dr. Amanda Marinoff, who is attending pediatric oncologist and clinical instructor at UCSF to talk about it. I want to say that we are, just a few weeks ago, I had the opportunity to be at the FDA and was talking to Dr. Peter Marks about, who's the director of the Center of Biologics, whom I know fairly well. And Peter was talking about how the science has improved so much in the last, he said specifically the last 5 years. And then I was talking with Jeff Shuren, who was also at the same meeting that I happened to be at, who was the head of CDRH, the device division. He was talking about what's happened with all of the device indications that are being brought forward right now. And so innovation in science has really just blossomed over the last 5 years, and enough so that Peter said he had only 104 projects right now that are in the gene therapy area that are waiting to be approved. And his problem was he didn't have enough people in the organization to be able to get them through the project. So I think today's panel will has some vignettes and some stories they want to share with you all. So I'm going to turn it over to Dr. Shuster, and thank you all for being here this afternoon. And remember to get your ticket packet.
Thank you, Peter. And without further ado, why don't we go get to beginning? Amanda, would you like to be the first? Oh no, you're teed up for doing stuff, Don. Go ahead.
Okay, can I have the first slide? I'm going to show some slides, and then I'm going to show you a very cute patient after that in the video. So I'm at UCLA, and I just want to welcome everyone to Southern California. And you've probably by now all learned the term June gloom. This is our refrigeration, keeps our tomatoes very happy in the early summer, but it will be sunny later today. So I'm going to talk about work we do on gene therapy for a specific set of diseases, blood cell diseases, so immune deficiencies, red cell diseases like sickle cell, some lysosomal diseases. And I'll talk about one in particular that we've been treating. Then we'll meet a patient who was treated for it. And so the disease that I've worked on is called severe combined immune deficiency, or SCID. It's also known as boy in the bubble disease. This is where children are born with no immune system. Whoops, we're advancing on our own. And so a little biology here. So in our bone marrow, we have hematopoietic or blood-forming stem cells that every day are turning out billions of new red blood cells, white cells. But one cell is special. It needs to go to college first. So to make a T cell, it has to go to the University of T cells, which is an organ in the chest called the thymus. And it actually learns there how to recognize foreign things and not attack self. And so those then go out into the blood and do their job. And in this disease, SCID, that doesn't happen. So patients have no T cells to fight off viruses, funguses, and so typically before there were treatments, they would die in childhood from this disease. And we know that, in fact, if we take hematopoietic stem cells, these blood-forming stem cells from a healthy, often a brother or sister who's a match, and transplant them into the patient, we can cure a whole list of genetic diseases. And so there are primary immune deficiencies like SCID that I talked about, but a number of other ones where one gene is missing needed for the immune system to develop, or sickle cell that many people are more familiar with, where there's a mistake in a gene for hemoglobin, or other diseases. Because these are all blood cells, if we give them back the mother blood cell, the hematopoietic stem cell, from a healthy donor, they can make normal blood cells. And so transplants for these diseases from brothers or sisters or unrelated have been going on for about 3-4 decades. And although it's certainly a major medical procedure, they can be curative. And the main risk is that the donor's cells can react against the patient and cause an immune response. And so the approach that we're taking, and a number of other people are to take the patient's own stem cells. We can collect them from the bone marrow, for example. In the laboratory, add a normal copy of the gene that's missing. And once we put that into the stem cells and give those cells back, for the rest of that patient's life, those stem cells are now genetically normal again. You can make the needed blood cells. In SCID, that would be T cells. And so we did this for this disease that I'm talking about, SCID, and a certain subtype of it, a gene called ADA, or adenosine deaminase. So we did it in parallel with colleagues in London, so Great Ormond Street Hospital and UCLA Mitchell Children's Hospital. And we published our results a few years ago from the first 50 patients treated, 30 at UCLA, 20 in London. And we pooled our results together. And what we reported was that 100% of the patients were surviving from this otherwise fatal disease. And in 48 out of 50, it worked very well. And so they had the stem cells that we took out, grew back, and are making a good immune system. 2 patients, it didn't work, and they needed to have a transplant from a donor. And so this is just one piece of data where we're writing up a paper now. Where are they now? Because we're out 10 years on this group of patients that we published earlier. And it's just flat stable. So the T cell numbers and all the different immune cells that you're seeing here, the numbers of those are stable in all these patients now out to 10 years. So we're hoping it's a permanent treatment for this rare disease. And so that's kind of the good news. And as we heard, you know, the science is good, and we've really made so much progress for a number of diseases. The problems really are becoming more financial, regulatory. And so we applied for this therapy for SCID for all the FDA gold ribbons of orphan disease, breakthrough therapy, and rare pediatric disease designations. And in fact, almost now, I guess about 8 years ago, we licensed it to a companies, because once you've in academia shown a therapy can work, it needs to get commercialized and become a drug to become available in our current system. So a company, Orchard Therapeutics, licensed it, and then after 4 years they decided they weren't going to continue to move forward with it, and they gave it back to us. And so between 2018 and last year, no one got treated for this disease, and in fact we accumulated a list of 30 patients waiting for it. And so once it came back, we are funded by the California Institute for Regenerative Medicine, CIRM. That's our stem cell initiative. And so there was funding remaining, which came back to us. So we're treating patients under it compassionately, but we're really working very hard to try to find a way to get a sustainable approach for it. And so I'll stop there, and then I think we have a video to see a patient who got treated about a year ago for this disease.
You are. Say your name.
Shersha.
And how old are you?
6.
6. And I'm Shayla. I'm her mom. And she has SCID. So she was born in Hawaii. And when she was 6 days old, we basically got a phone call saying that there was something wrong with her T cells. And I do not recommend Google. It was the very first thing I did. And there was a whole host of things that were wrong. And I was like, that was not a smart idea. So when she was 6 days old, we were air flighted on a KC-135 medical flight from Hawaii to LAX. And we didn't know like what was gonna happen. My husband had to stay home with our other children because he was stationed there. And it was one of those like most terrifying moments, because it was, is he gonna see her again? She can't come back to the island, and we didn't have a formal diagnosis, like we weren't sure what was happening. I actually had my sister, who was at the time at Cal State Northridge, and she came and met us at the hospital, and she took notes that day for us, because my mind just was not in the place to do it. And she spent 55 days in reverse isolation the first time. And we took her home when she was about 4 weeks old. And it was, and we had to take her home with a bunch of medications. I was taught how to do infusions and shots in her legs with her medications. And I've had no formal training. So it was just like, I have a business degree. I was like, I was not mentally prepared for this. And my older children were in school, they still are. So it was, we brought her home at my parents' house because we couldn't go back to Hawaii, and we had to move from Hawaii back to California for this treatment, basically. And they were basically like she was stable on Revcovy, which is a medicine that ADA SCID is able to use. It's one of the better parts of having ADA SCID, I guess you could say. And we had all this testing done. My 2 older siblings, they're not matches for her. My husband and I were not even half matches for her. So when we took her home, we knew that she didn't have any matches, but she could do this medication called Revcovy temporarily, basically, to help her have her Band-Aid immune system. And she had, we actually met Dr. Kohn and his team, and they were like, had she been born anywhere else, gene therapy wouldn't have been offered to us at the time in 2017. But because the clinical trial was at UCLA originally, they were like, you could wait. So we decided to wait for it, thinking it was going to be about 2 to 3 years, wait for her. And we took her home and we kept her isolated. She had her own room, a clean room. My poor children, when they would get sick, it was basically bathed in Lysol, and you have to go to your room. You can't leave your room or your bathroom because she couldn't get sick. If my husband or I ever got sick, we had to sleep in a different room, and we were very cautious of making sure that she didn't get sick. And she kind of has always grown up that way. So I don't think she really noticed a difference until she was probably about 2 or 3, that she wasn't normal. And my husband and I hate that word. We used to like the word normal, and you pray for a healthy baby, you pray for normal, and then we kind of realized that nobody's normal, and who cares? So having her definitely changed our mindset of what normal meant, because who wants to be normal? And so my older kids would go to like Disneyland or a Dodger game, and she would stay home with me. And she was just used to it, basically. And that's why her dream now is like, when she's cleared, she must go to a Dodger game. Yeah. Where else do you want to go?
Disney World.
Disney World. Who's your favorite princess?
Aurora.
Yeah, so she wants to go to Disneyland, Disney World, do all these things. Up until recently, she's been like sequestered to like our house and things that are near us.
Where is Aurora?
She's at the castle.
On the Elsa Castle?
No, they have their own castle.
Where?
Disneyland.
Oh.
And in 2020, she was set to start her clinical trial, but COVID happened, and then the pharmaceutical company shut down the trial. And so we were devastated. We were terrified too, that we had already waited 3 years for this trial, and it was kind of closed down. And I remember calling her team and saying, what next? Like, what do we do? We've waited. What's next? And Dr. Kohn was like, this is my life's work. I'm gonna figure it out. And we put all that trust and faith in it. And we got the call about, 2022, the end of 2022. And we would start our workup in January of 2023. And we did all of the workout for it. And then she had her stem cells removed in April. And then they were given back to her in May. And so she's 1 year post-transplant. And while she was in the hospital, she played with everybody.
Doctor.
The doctors, the nurses.
And she gave me Play-Doh.
They gave you Play-Doh. What else did they give you?
Peppa toys and with the swings.
And what else?
Toys.
And Barbies. How many Barbies did you bring home?
4.
More than that, probably, huh?
5.
Yeah, she brought home a bunch of Barbies. She played with everybody.
The doctor.
Everybody played with you, huh?
And I brought Play-Doh. I still have the Peppa Pig glasses, so.
Yeah. And so now we're 1 year post-transplant, and she is about 500 T cells. She was born with 5. Before she got her transplant, she had 56. So we're definitely moving up in the numbers and things. And she's homeschooled, so she can't go to school yet. She can't have vaccines. There's been a lot of sacrifices I feel like she's made, but she's the most joyful person I've ever met. and she brings light to everybody she meets. When she's in the clinic, everybody's excited to see her. When we were in the hospital, like, people who'd never played with kids before were playing with her because she just brings that kind of energy. And so she changed my husband and I's life. Like, we have 2 healthy children, and she was our tiebreaker. And the adjustment period from moving for her and sacrificing everything that we needed to do for her, and yet she's the best thing that happened to us. Our children are great with special needs children. They're great with people who are a little bit different because they see it at home, and I think that it makes them better people for it. And she has never met a stranger. My husband and I have joked repeatedly that she is the type of child that would get into a van if you had puppies or Smarties, or because she's never met somebody who couldn't be her friend. And I don't think that our life would ever be the same without her. So to the parents who have a new kid and it's the most terrifying thing in the world, that it gets better. We had 6 years of terrifying, and even now, I don't think I'm ever going to be okay when she goes to public school, honestly. But she changed what normal is for us, and we love that.
Amanda, follow that up.
Well, that is an impossible act to follow. It is such an honor to be here and to talk with you all about the landscape of pediatric cancer and some innovations that are really transforming it for the better. I'm going to take the rare opportunity to go au naturel or without slides today to have this conversation. I wanted to first start by saying that all pediatric cancers are rare, but still it's too common and is the leading cause of disease-related death in children. Our patients often undergo very long and toxic therapies, including a number of modalities, including chemotherapy, radiation, and surgery. And these have side effects in both the short and long term. And while we've made very significant progress in the last few decades, we still are not able to cure every child, and until we're there, we still have work to do. But today I wanted to highlight 3 innovations that really offer hope that we can and will get there. And the first is precision medicine. For those who were at the former breakout session on genetic testing, I think there's a lot of themes here that overlap. Advancements in our ability to sequence tumor cells, both the DNA and now the RNA, have allowed us to identify the specific driver mutations or changes in the genetic code for each individual patient and allow opportunities to tailor our treatment for each individual patient. We can also sequence the non-cancer or healthy cells and determine whether the cancer was caused by an inherited cause or was sporadic. And that can have implications for other family members, our screening processes, as well as treatment in some cases. Sequencing the tumors also allow us to gain information about how well a patient is likely to respond to therapy, what their survival may be like. And that can help us to work toward what we call risk-adapted therapy, where the goal is really to hit a Goldilocks spot such that we give enough treatment that we can cure a patient, but not so much that we're exposing them to unnecessary side effects. In my own translational research, I focus on osteosarcoma, which is a rare and aggressive bone cancer that primarily affects adolescents and young adults. And the treatment for osteosarcoma has really remained the same for the last 4 decades, as have the outcomes. And treatment for this cancer is a one-size-fits-all approach right now. And my work focuses on identifying genetic biomarkers to help us really figure out which patients are likely to respond to our standard therapies and which patients are likely to go on to relapse and therefore need new and different therapies that are more tailored to their biology. And the goal is to translate these biomarkers and novel therapeutic strategies into clinical trials that will ultimately improve outcomes and decrease treatment-related toxicity for our patients. The second innovation that I want to highlight is immunotherapy. And that is essentially sort of a broad category that says that we're harnessing the body's immune system to go after and kill cancer cells. There's a variety of ways that we can do that, and researchers are working actively on expanding the options for that, as well as the people who may benefit. CAR T cells, or chimeric antigen receptor T cells, is a type of therapy that has really, for a subset of patients with leukemia that has come back or is refractory, really revolutionized outcomes. And to make CAR T cells, we take the T cells out of the patient's body, engineer them in a lab to go after and kill the cancer cells, and then reinfuse the T cells back into the patient's body. Not so dissimilar to the stem cell therapy that Dr. Kohn was talking about. I just want to highlight a couple of other types of immunotherapy. They don't all involve taking out the patient's immune cells and turbocharging them. We can actually give medications in some cases that really activate the patient's immune system within their body to go after the cancer cells. And you'll hear soon from one of my patients, Wavy, who received a type of immune-based therapy called blinatumomab, or Blina, which is a bispecific T-cell engager that takes the body's own T-cells and tells them to go after B cells, including the B-cell leukemia cells. And treating Wavy with blinatumomab allowed us to not only effectively treat her cancer, but it allowed us to do so in a way that did not significantly dampen her own body's immune system and concurrently fight a life-threatening fungal infection. And previously, those two goals were thought to be impossible to tackle together. You'll hear Wavy's story in a bit. And the third area of innovation that I want to highlight is research into understanding and tackling mechanisms of disparities in cancer outcomes. We know now that socioeconomic status, race, ethnicity, where you live can all impact cancer outcomes. And researchers are now rigorously researching the mechanisms. Are there genetic or biological bases to that? How does that interact with access to care, to clinical trials, to desire to get care? And this is extraordinarily important for ensuring that our innovations in cancer care are being brought to every single child and family with cancer, regardless of where they live. And so in closing, I think these are just three of the innovations that give me a lot of hope. I often say that I have the best job in the world, and I really mean it. And I think the progress that we're making in the field and the pace of it really helps to drive that. As do my patients. They are what get me up in the morning and sometimes keep me up at night. And so with that, I want to introduce you to one of my patients, Wavy, and her parents, Mark and Ramona, via video, and I'll turn it over to them.
Hi, I'm Ramona Altman.
And I'm Mark Altman, and we're here to talk to you about our daughter, Wavy. In December 2020, Wavy was diagnosed with leukemia, and we figured this out with our regional hospital, and they assured us that it was treatable, and we knew that we thought things were going to be okay. But after about a week of being there, she ended up getting a fungus ball found in her lung, along with a ring of fungus around her lower eyelid. And while the doctors assured us that they could also treat that, Wavy started to get a lot worse. She wasn't eating, and she was getting very high fevers again. And we were worried, very worried. And so we decided the next best thing to do was to take her to a university level hospital like UCSF. And we're very happy we did. But when we got there, it was January 1st of 2021. And after about 6 or 7 hours, the doctors there told us that she was not going to survive. And it was extremely upsetting, and we felt like our lives were over. And
The fungus had spread through numerous organs, so it just wasn't in her lung and in her eye, but it had gone through her spleen, her kidneys, like we mentioned, her lung, both lungs, and liver,
Yes,
I believe.
yes. And we couldn't believe what was going on. I mean, a few days earlier, the doctors had let us know everything was fixable, and then all of a sudden we were being told that things really looked bad. And so we hunkered down and we stayed at UCSF hospital for over a month, about a month and a half. Mm-hmm. And during that time, the doctors told us that it was going to be a miracle she survived. They said it was out of the realm of medicine and it was totally going to be a miracle. And we decided to do everything we could, and we tried our best, and we thought about everything, and the doctors were right there with us. And so we were really impressed with everything that happened, but every day was difficult. It was middle of COVID. We had 2 other kids that weren't able to be there, but Ramona and I stayed in the hospital with Wavy, and it was a very upsetting time. But the good news is UCSF has the best staff, the best doctors, and they did everything. They thought so deeply about Wavy. They were tireless, and they came up with solutions and things that they had never even thought about before. They went and tried. And
they also listened to our concerns and made sure that what we wanted, worked with what they were thinking of in terms of treatment and, quality of life for Wavy.
I mean, they were just so thoughtful. First thing they did, doubled her dose of antifungals.
Took her off of chemo.
Stopped her chemo. Took a biopsy on the eye, which we thought was going to disfigure her eye, and it didn't really do much to her eye. We were thrilled about that. Even though we didn't think she was going to survive, we thought, wow, they're really even thoughtful about that. And so time went on, and after about a month and a half of us crying, panicking, being completely upset, they said, we think she can go home for a week. We were both happy and scared at the same time.
At that point, she was on so many medications, and they had just decided to put her back on, I don't know if I'd call it a chemo, but it's kind of a chemo. They put her on Blina, which she had to be on for 30 days nonstop.
The doctors walk this tightrope of, we have to cure the cancer, but we got to cure the fungus. And Blina held the cancer off while the fungus was being taken care of. And day by day, Wavy kept getting stronger. But throughout that entire year, Wavy was taking food through a nose tube and throwing up all the time and not looking good a lot, and us thinking a lot of that year that she was not going to survive. But as time went on, she kept getting stronger. We spent about half of the year, I would say, in the hospital on overnight visits in 2021.
On and on.
And
some planned visits, some unplanned.
Wavy was a trooper, and we advocated for her all the time. And the parents are are the kid's best advocate. But the amazing thing is that the UCSF doctors and staff were right there with her. They were like family. They treat her so well. They treated us so well, nurses so well. It was incredible. And as the year ended and then 2022 started, we made more and more progress between regular chemo, between the Blina, between the antifungals. We did it, a lot of it we did at our house by ourselves, with Ramona being the primary caregiver and taking care of Wavy. And as time went on, hope kept building more and more and more. And we got to a place where we started to get more comfortable. It took about 2.5 years, and Wavy in the middle of 2023 ended up getting her nose tube out so she could start eating normal food.
She was going to school.
Starting to be like a normal kid and enjoying her life and letting us relax a little bit, but not really relax, but a little bit. And knowing that what we did and what the doctors did, while it was so painful and so hard and so saddening, it ended up turning into an amazing miracle where she survived, and now she's thriving, and we're thankfully thriving as a family together.
She's completed her treatment. She got to ring the bell last summer.
Yes.
She just finished first grade. She's doing dance. She's doing swim lessons. Just being a normal kid.
Yes. And so I think I speak for both of us when I would say, first of all, thank you, UCSF, the best doctors and staff in the world that did everything imaginable to get our child to survive. But the things also we would say is, number one, if you're in the same situation, you have to take things day by day. It's so easy to go to a dark place and think the worst about things. But if you take things day by day, it'll put your mind at peace. It puts, I think, the child's mind at peace, so that they have the best possible chance of survival. I would again reiterate that you're the child's best advocate as their parent. You are the one who knows your kid. You are the one who is able to think for them and speak for them and really just go to bat for them. And so you need to make sure that you are taking that as seriously as possible. And the amazing thing is when you go to a place like UCSF, they treat that responsibility just as well. And they go after it just as hard as we did. And it was just such a team effort and such an amazing place. And I don't know how we would have done it without them. I think it was just the most, we're so thankful.
I think that was the best decision we made throughout this whole thing, was transferring Wavy over to UCSF.
Yes. And so, to end, we're gonna bring Wavy out. Waverlyn, can you come out here to come say hello to everybody? Here she comes. All right, here she is.
Here she is. This is Wavy. Wavy, say hi.
Hi.
How old are you?
7.
7. Is there anything you'd like to say?
Thank you UCSF for helping me and taking care of me.
Good job.
Yes, and thank you UCSF from the Altmans, all of the Altmans, even from us to extended family to our other children. Thank you again. The best doctors, the best staff, everything the best. We love you all. Thank you so much for saving us and saving our daughter.
Say thank you.
Thank you.
Bye.
Bye.
There's Glenna outside of UCSF, to be clear.
Marketing delight.
Yeah. I mean, one of the things I don't want to do is get in between two UC campuses here to decide where the best doctors are.
But they don't have a football team.
That's true. You got a miserable football team.
The weather's a lot worse, too.
But with that, thank you both for those incredible talks. And I think what to me is so inspiring is the work you're all doing, and it ranges all the way from the bench to the bedside to the community. And to me, that's an incredibly powerful tool and a message, and it should give us all an incredible amount of hope that there are so many good people working so hard in LA and San Francisco to work together with the rare disease community. And I just want to thank you too. But I know there are questions, so it's time now for questions.
Great,
Tiffany.
thank you. How can families become involved to further advance breakthrough therapies in their own disease states?
Yeah, I'm happy to start. Thank you for that question. I think as Mark and Ramona highlighted, I think being your child or family member's best advocate is sort of number one with their clinical care. But I think in terms of advancing breakthroughs, we learn by having tissue or samples that we can genetically test and learn about the biology from. And so we are where we are in terms of our progress because of the generosity of our patients in contributing to research by sharing their tumor samples, their healthy samples, undergoing procedures and donating and saying, we want to learn more, we're hungry for that, and please do whatever you need to do is what many of my patients share. And because of that, we have what's called a tissue bank or a biobank of patients' samples that allow us to really rigorously study sequence at multiple levels, develop animal models that are specific to the patient's cancers. And these are the ways that we're going to learn more about biology and to drive breakthroughs and hopefully more cures.
Yeah, I would just add to that, clinical trials, all the kind of studies you're talking about are done in that context, and that's how we learn in medicine, is to do a formalized study of a new therapy versus the old therapy or whatever. Doesn't mean blindly take any trial. You know, when we talk to patients about being in trials, we have an informed consent document. We spend a lot of time really trying to explain clearly risks, benefits, potential upside, potential downside. And so you need to consider whether it seems right, but it is really the way that we learn, and all the therapies that we have now came through trials over the years.
Yeah, and I want to echo that. I enroll many of my patients on clinical trials, and I can only imagine how scary it is to be in the other seat, sort of where you're enrolling on a study that is an experimental question, and the doctor comes in with a large packet of paperwork to do that informed consent process. And if I were in that position, I might be shell-shocked. What do you mean? And you want me to participate in an experiment, and I have to sort of, what am I signing away? And I would just encourage you to ask questions. The consent is to keep everyone safe, and is really sort of a door opener for participating in the clinical trial, but I think can be really intimidating. And I think just want to say it's a huge part of advancing the field, and please ask questions. There's no reason to feel the sort of the discomfort of the big packet and the complicated words around it to be a barrier.
Great, thank you so much. If we live in an area far from a trial, how do we get enrolled in a trial or get our samples to one?
Often there are specific studies. You can go on clinicaltrials.gov where there are specific centers that run a clinical trial, and many times that is, I'm acknowledging it's sort of a privilege to be able to get to one of those centers, to sort of uplift your entire life, leave other family members' work behind. And so I think, one, acknowledging that it's not perfect. We want to make clinical trials accessible to every patient. One way to help bridge that gap is to work with your local center's social work team to find out ways of, and the clinical trial center's social work team to find out ways that we can support patients in traveling to far-flung places, get you set up with housing and other resources while you're participating in a trial. I've certainly had patients from many states come to UCSF, and I've also sent many of my patients working closely with both social work teams to allow patients access to trials that we think can be helpful, and to have sort of place not be a barrier.
Okay, thank you.
Yeah, I agree. That you have to advocate for it and find a way to get there. You know, learn about them. Clinicaltrials.gov, again, is the best site that has quite accurate information about the trials. And if there is one that really seems, you know, for the disease you're working with, you have to just push for it. And again, social workers and others can help often find the resources.
My patients are often shy about asking about a clinical trial or even a second opinion at another center. And I always let them know I have no agenda other than make sure you get the best possible care that we together feel as a team. It's not about me, it's about you all and your child. And so I think if you have the thought in your mind, I want to maybe go elsewhere, explore elsewhere, please have the courage to ask your provider, because we're all a team, and that's just an opportunity to expand the team.
Great, thank you. You shared 2 success stories where complex rare conditions were treated with wonderful outcomes. Do today's successes have implications for other rare disease patients in the future?
Yeah, well, certainly in the gene therapy area, I'm sure many of you know about gene therapies in the diseases that you're interested in. So there's great advancements occurring for, you know, just hundreds, literally, of diseases where there will be, you know, the logic of gene therapy is if the gene is the problem, how do we fix it? And the tools that we're developing are growing increasingly sophisticated. So I talked about sort of version 1.0, where we put the gene in with a virus, which can be useful in some places, but sort of 2.0 and beyond are things that you may have heard about with CRISPR, which is an enzyme that can actually go into the whole genome and fix one specific site. And there's all these variations now from CRISPR, base editing, prime editing, on, that are just really precise at correcting mutations. The challenges for many conditions, how do you get it to the right site? How do you get into the brain, the liver, whatever organ's involved? We have the advantage with bone marrow, we can take the cells out and do that in the test tube. But the same kind of technology that's been used to deliver, like the COVID vaccine, lipid nanoparticles with RNA, is coming along, again, to deliver genes or gene correction tools throughout the body. So I think there's just, hopefully the next decade, we're going to see a lot more cures for many diseases.
And I'll add, I think there are sort of two elements of Wavy's story that could be applicable to other patients. First, immune-based therapies are being utilized for lots of different diseases, including autoimmune and immune dysregulation and autoinflammatory disorders. And so I think that's going to be more broadly applied outside of the context of cancer. And then what Wavy's family didn't share was that we actually tested her leukemia cells for the underlying driver mutation. And in her case, it was a rearrangement called ETV6-RUNX1, which we knew was associated with a favorable prognosis. And that gave us the courage and the data to pursue an approach where she was able to receive sort of lighter therapy for really throughout her therapy. And I think had we identified a much more aggressive prognostic factor, we would not have been able to do that. Our strategy would have been really different. And so I think in many rare diseases, genetic testing, as we talked about in the last breakout session, can be really informative for helping to get diagnostic clarity and really to tailor a treatment strategy to the underlying biology.
Great, thank you. How do you know when it's time to transfer care to a research or university hospital, and what can a parent do to advocate for that?
You want to take that one?
Go ahead and take it.
It's a very difficult question to answer because it's hard to know when that is. Those of us who are at sort of tertiary academic medical centers, we think we know everything. We don't. But there is often, much more deep knowledge in teams that take care of these disorders. And so, I think in general, it's good to be seen at least some point early on at one of those centers, even if you're going to get most of your care locally. And the NIH, for example, has the capacity to bring patients all the way to Bethesda, Maryland. And so in some cases, it's useful to be seen sort of by the real experts in the disease. And often, again, the treatments can be developed and applied, given locally.
Yeah, and I think if you have more questions than answers, that might be a cue to seek additional opinions. Recognizing, again, academic universities don't know everything. I think in the space of rare diseases, there often are more questions than answers, and often we can sort of bridge the care and partner with a larger university and a local hospital to together sort of create a larger team that also works for your life and your family.
That's a great point. Unfortunately, we are at time, and there are a couple of questions that we weren't able to get to. So once again, I feel that they're very welcome to be approached afterwards if you would like to get those questions answered. Thank you all.
Thank you, NORD.